In this lesson, we'll look at the phenomenon of neurogenesis more closely. We'll see that there are sort of two sides to neurogenesis. Neurogenesis may be interfering with previous memories, but also preparing the hippocampus for the allocation of cells to new memories. Now, in another lesson, we learned about neurogenesis in the hippocampus. It's the creation of new neurons by cell division, taking place in a few regions of the brain. The hippocampus is one of those regions, so new neurons integrate into the dentate gyrus. Now, the dentate gyrus cells, here's in this part of the hippocampus, send axons over to cells in the CA3 region of the hippocampus. So they'll be synapsing on CA3 cells, and those axons are called the mossy fibers. And we encountered neurogenesis in the context of the question, why does the brain undergo memory consolidation? Why are memories reorganized across brain systems? That's what we call system consolidation. Why do that? Well, in that lesson, we talked about three possible reasons. Interference due to new learning that happens in the hippocampus, interference due to new neurons, and that's neurogenesis, and then interference due to synaptic resetting during sleep. So for all of these reasons, the idea is consolidation would be a way to get around the interfering effects of these three processes, that the memory will be transformed in a way that these things will not permanently disrupt the memory. So in answer to the question, why consolidate, one of those is because neurogenesis can interfere with memory traces in the hippocampus. So if neurogenesis interferes with hippocampal memory traces, then remembering is possible if system consolidation outpaces the interference due to neurogenesis. Forgetting will occur if neurogenesis outpaces system consolidation. Neurogenesis, in addition, may account for infantile amnesia because neurogenesis is significantly elevated during infancy in many species. So in this lesson, we're going to take a look at some evidence that neurogenesis really does interfere with prior memories, and then in part 2, we'll look at how neurogenesis may be preparing the hippocampus for future learning. Now, the training paradigm that will be used in this research is the contextual fear conditioning paradigm, where rats are placed in a certain context, like KJ, and given a shock. Rats are then returned to their home cage. If placed in that same training cage, rats will show a fear response. They will freeze for a period of time. Rats will show a fear memory to the context associated with the shock, but not a different context, for example, cage B, that was never associated with a shock. And in other lessons, we've talked about the important role of the hippocampus in this kind of contextual fear conditioning paradigm. The conditioning of a tone with a shock seems to rely on the amygdala proper, whereas the fear memories for a certain context requires an intact hippocampus. And so the question will be, does neurogenesis interfere with learning this kind of association, that a shock occurred in a certain context? All right, in the rest of this part then, we'll look at some research done by Akers and her colleagues. Now, first the pictures at the top. What the scientists are going to use a staining technique to stain newly born neurons. So this will be how they can visualize the magnitude of neurogenesis. And the animals used here will be mice, and P17 would be postnatal day 17, P60 would be postnatal day 60. So the adults are the P60 and the infant mice are the P17. And the idea is that if you simply just stain the brains of infant mice and to try to capture the magnitude of neurogenesis, you see lots of neurogenesis occurring in the infant mice. So lots of stained cells, lots of new neurons are being born in the hippocampus. By contrast, in the adult mouse, we have less stain here, indicating less neurogenesis occurred. Fewer new cells were being born in the adult. So neurogenesis is high in the infant and lower in the adult in mice. Now, with this difference in mind, that the infant mice show higher levels of neurogenesis than adults, they went on to carry out the contextual fear conditioning paradigm. So here we have the P17 mice, the P60 mice. They're going to be trained in a certain context, so they're going to get a shock in a context. Then they're going to wait 1, 7, 14, or 28 days. So that's the train test interval. And then the mice will be tested, simply putting the mice back into the context and see if they show fear behavior like freezing. Here we see the data over here. Let's take the adults first, the P60 in blue. Notice then, when tested 1 day after training, there is a high percentage of freezing here, so that indicates that the memory is strong, and in fact you don't see any decrease in the strength of the memory in a sense, right? So we have a constant level of freezing here, no matter when tested 1, 7, 14, or 28 days. The memory is being maintained over this time period. By contrast, for the infant mice, we see significant levels of freezing when tested 1 day after training, but notice a dramatic reduction in freezing behavior as we go to 7, 14, and 28 days. Now, why would we see this drop-off here in the freezing behavior? Well, Akers and colleagues are going to argue that this is the result of neurogenesis. So the high rates of neurogenesis for the young mice, there's a lot of neurogenesis going on in this time period, and that may be interfering with the memory trace in the hippocampus for that learned association. And so as time passes, with more and more new neurons being inserted into hippocampal systems, the memory, the fear memory, seems to be decreasing. So mice are trained in a contextual fear conditioning paradigm. The adult mice showed high levels of freezing at all train test intervals. Infant mice showed high levels of freezing only when tested 1 day after training. The more neurogenesis between the training and testing, the more forgetting. And this suggests that high levels of neurogenesis interfere with the maintenance or access of a fear memory. So at this stage, it could be both, that neurogenesis is actually interfering with the memory trace in the hippocampus or the access to that memory trace. Now, the researchers went on to see if they could manipulate neurogenesis, and other labs had discovered that exercise triggers neurogenesis. So Akers and colleagues are going to use a voluntary running procedure, and they in fact found that voluntary running does indeed induce neurogenesis in the adult mouse. And by 7 days, that increase is significant. So let's take a look at the pictures. Here we have in the dentate gyrus the picture of the sedentary mice on day 0 and the running mice. And so we get a sense of the rate of neurogenesis occurring here. Now for the sedentary mice, after 28 days of just being sedentary, this is what the dentate gyrus looks like when stained, and it's very similar. So we don't see any increase in the rate of neurogenesis. And the story is similar for CA3. But look at the running condition. So again, on day 0, we have a certain level of neurogenesis happening in these regions. Day 28, after 28 days of regular access to exercise, voluntary running, we see a lot more staining here in both dentate gyrus and the CA3, indicating higher rates of neurogenesis. So there are more neurons being born in the mice that had access to some exercise. And down here we see the data, and you can clearly see the pattern that the running condition is elevating the number of cells that were stained. Also the large mossy fiber terminals increased as well. So now that it's established that the voluntary running can induce neurogenesis, they wanted to see whether the running would have an impact on the memory. So let's look at this diagram up here. We've got, again, the group of sedentary mice and the running mice. They're all trained on this day here. The sedentary mice for 42 days, so 6 weeks, they're just not doing much. And then the other group are going to have access to a working wheel here, so they can do their voluntary running. And in this group, we would predict that the running should be stimulating neurogenesis. The question is the neurogenesis going to influence the retention of the memory across this delay? First, over here we see the cells, again a confirmation that in the running group, the blue bar here, we have more cells that were stained, so we seem to have the neurogenesis was triggered by the running. So we've got more cells. The question is, what happens when the animals are tested? They're put back in the chamber, and we measure freezing behavior, and lo and behold, the animals that were in the running condition show more forgetting. So in other words, freezing levels are reduced compared to the sedentary mice. So this suggests that the neurogenesis that was triggered by the running here is interfering with the original memory of this association, the shock in a certain context. We're getting significant forgetting in the running condition. Now, if this wheel is locked, so it's in the cage, but it's locked, it doesn't work, then you do not see this decrease here. So it's not just novelty that is the effect. It's the exercise that seems to be triggering the neurogenesis that is interfering with the memory. Now, interestingly, they went on to reverse the order of the exercise and the training. So take a look at this paradigm down here. We've got the two groups, the sedentary and the running mice. The sedentary mice are just sedentary for 42 days. The running mice are able to do exercise for the 6 weeks here, and then they're trained. So they're both going to be trained after a period of sedentary behavior or exercise. So then they're trained and then 1 day later tested. So notice then neurogenesis is happening before the training here. Again, the data shows that neurogenesis did in fact produce more new neurons there. So we get the blue bars higher than the white bar here. But notice for the test of the memory, the freezing behavior, we get no difference. In other words, even the animals that were running and had neurogenesis happening more so than the sedentary mice, those mice still retain the memory. The neurogenesis did not interfere with the memory. And of course, the explanation here would be because the neurogenesis had nothing to interfere with. The training happened after the neurogenesis that occurred in this 42-day period here. So adult mice were trained using a contextual fear paradigm. Some mice remained sedentary for 6 weeks, while others had access to a running wheel. When tested after 6 weeks, mice that had running-induced neurogenesis showed greater forgetting compared to sedentary mice. That's up here. Locking the running wheel eliminates this effect, so it's not due to novelty. So if you lock the wheel, it abolishes that effect. When the running manipulation occurred first, down here, neurogenesis increased, but there was no effect on the fear memory, as shown by the bars here. There was no memory for the new neurons to interfere with. So the neurogenesis has to be happening after some learning event if it's going to be interfering with the memory for that event. So now while running can be used to induce neurogenesis, and there are other pharmacological techniques to induce neurogenesis, the scientists went on then to see what would happen if they blocked neurogenesis. So can procedures that block neurogenesis prevent the forgetting that was found in earlier experiments? So here we have the procedure. Again, we got the two groups of mice. We got the sedentary mice that were trained, and then they are sedentary for 6 weeks. We have the training here, and then the running exposure to the wheels. So we got neurogenesis happening here, and then testing. So we've seen that this is the earlier research. We know that there's an increase in neurogenesis in the running condition, and that leads to forgetting. But now what happens when you block the neurogenesis that was triggered here? So researchers are going to do a pharmacological technique to block neurogenesis in both groups, and let's see what happens. What happens is there's an overall reduction in neurogenesis in both groups. And notice what happens to the memory. We now recover the memory. So by blocking neurogenesis, the animals do not show the forgetting anymore. So in the running condition, previously they were forgetting the memory. Now, because neurogenesis had been blocked, they are showing similar levels of freezing behavior. So this suggests that neurogenesis really is interfering with the memory traces in some way, and that by blocking then the neurogenesis, you can prevent that interference and the memory can be retained. The researchers then went on to exploit some genetic differences between two species. So mice show high postnatal neurogenesis and high infantile amnesia. Memories decay in infancy. So we've just seen that data in mice. Infant mice will quickly forget a contextual fear memory. And the suggestion was it's because they have high levels of neurogenesis. By contrast, guinea pigs show low postnatal neurogenesis, and they also show low infantile amnesia. Memories persist in infancy. So again, this supports the idea that neurogenesis is sort of correlated with forgetting in infancy, well, one explanation would be neurogenesis is causing the forgetting. And the guinea pig has low neurogenesis, and so you're going to have fewer neurons interfering with previously established memories, and so you'll get low infantile amnesia. Now, they took these two species then, and they tried to manipulate the amount of neurogenesis occurring. So for the mice, you can reduce neurogenesis, and when you reduce the neurogenesis in mice, they start to look like the guinea pigs. At least the behavioral data starts to look like the guinea pig. When you take the guinea pigs and you increase neurogenesis, that makes them more like mice. And so this was a nice addition to their study here, that using two genetically different species that show different levels of neurogenesis, if you manipulate the neurogenesis in either species, you can make them behave more like the other species. Now, in other lessons on memory, in focusing on the hippocampus, we thought of the hippocampus as integrating information from high-level perceptual areas. And the hippocampus then will undergo synaptic plasticity among subsets of cells, and this will form a memory trace for a given event. So episode 1 has there are sounds and sights associated with it, perhaps a feel, and synaptic changes will occur here to make an engram, a memory trace for episode number 1. And the idea then is that at a later time, if any particular cue is presented that was part of episode 1, that cue can reactivate the pattern of hippocampal activity, and that would constitute retrieving the memory. So in this diagram, we see an engram, a memory trace for event number 1 is here, and event number 2 is here. And in this diagram, we're showing that a cue from episode 1 or event 1 is being experienced. Well, that will reactivate the engram or the memory trace for event 1, but not event 2. And so we'll be retrieving the memory for event 1. And this we said this was pattern completion, that the cue will reactivate that hippocampal memory trace. So in pattern completion, a cue that represents part of a past learning experience can reinstate the full pattern of activity. Now, here's where neurogenesis comes into play. Neurogenesis may interfere with pattern completion, making it difficult for the cue to activate the appropriate set of hippocampal neurons. After all, if we have new neurons being born and then taking their positions in the neural circuits in the hippocampus, we may be disrupting this pattern, this memory trace, so that then when the cue comes, the cue can no longer effectively or efficiently reactivate that set of cells. So neurogenesis may be physically interfering with memory traces in the hippocampus, making it difficult for cues to reactivate the pattern of cell activity that was present during the learning episode. So in summary, Akers and colleagues have shown, using a variety of techniques, strong evidence that neurogenesis is interfering with memory traces in the hippocampus. It's inducing forgetting. And when you increase neurogenesis, you can increase the forgetting. When you block neurogenesis, you can reduce the forgetting.